Today we'll be talking about genetic engineering. This is a topic that is included in the unit of biotechnology. Now, today's contents are what genetic engineering really is, how did it start, the process of genetic engineering, real life examples of genetic engineering, and benefits of genetic engineering. Mahdi, you can take over.
Okay, so what is genetic engineering? Genetic engineering is the manipulation of DNA to alter an organism's characteristic. The process by which pieces of DNA are transferred from one organism to another. It involves the group of techniques used to cut up and join together genetic material, especially DNA, from different biological species, to introduce the resulting hybrid DNA into an organism in order to form new combinations of heritable genetic material.
So, as we can see in the picture, it shows are how human goes to bacterium, human, and then fish. It like the transfer of fish to plant, human to sheep, bacterium. They're like showing us how common these two things are to each other and the DNA. So this is a video that will put the link that basically has a brief explanation about genetic engineering, and we'll keep the link below so you guys can watch it. Now, the history of genetic engineering. In 1972, there was this guy named Paul Berg, and he was the guy that combined DNA from the monkey virus SV40 with with that of lambda virus, which was the creation of the first recombinant DNA molecule. And in 1973, Herbert Boyer and Stanley Cohen they created the first transgenic organism by inserting antibiotic resistance genes into plasmids of an E. coli. We'll be talking about this more later on in this presentation. In 1986, tobacco plants they were engineered to be resistant to herbicides in the UK and France. Now, we will be talking about the process of genetic engineering, and I'll begin by saying that the first step would be the DNA carries a gene of interest, and this is taken from a human cell, and then the gene is inserted into the DNA of another host cell, and then this host cell it now contains recombinant DNA, and then the fourth step would be the host cell multiplies, and then the protein will be produced. Mahdi's gonna explain
So,
the procedure in depth now.
so basically, the restriction enzymes cut the desired gene from the genome. Respectively, mRNA can be treated with reverse transcriptase to produce short DNA segments. And then the same restriction enzymes cuts into the plasmid. Because it's the same restriction enzyme, the same bases are left exposed, creating sticky ends. So the ligase joins the sticky ends, the gene into the E. coli plasmid. A recombinant plasmid is inserted into the host cell. It now expresses the new gene. An example of this is human insulin production.
Okay, so moving on, I'd like to talk about genetic engineering a bit more and to give you guys a clear image of what it really means. Genetic engineering can also be known as genetic modification or gene transfer or transgenic. And I'd like to say that all living things they use the same basis and the same genetic code. Now, each codon it produces the same amino acid in transcription and translation, regardless of the species, and so the sequence of the amino acids in the polypeptide remains unchanged. Now, therefore, we can take genes from one species and insert them into the genome of another species. And some examples of genetic engineering would be milk or golden rice and human insulin. These are some examples that we'll talk about them later. on in the presentation. I'd like to talk about gene transfer, which is also called gene genetic engineering, and how it can also be used in gene therapy. Now, a virus vector is used to insert the recombinant plasmid into the genes of the affected cells, and the virus is chosen or designed to target only these specific cells. Now, examples I'd like to mention would be that recently hereditary blindness was treated with gene therapy, and it was a subretinal injection of vector technique. They used this certain virus vector, the one that I just mentioned, to treat hereditary blindness, and severe combined immune deficiency can also be treated this way. And this basically shows by gene therapy, to be exact. And the cells are intubated with a type of virus where the viral genetic material has been engineered to carry a normal copy of a gene. Now, uses of genetic engineering. As I said, insulin would be one. Now we'll talk about these. So, to make insulin of fuses for diabetic people, to make growth hormones, to treat dwarfs, to prepare vaccines, to make plants resistant to diseases, and to make pigs, cows, or fish grow faster, higher production of milk by cows. As I mentioned, that another example would be tolerant tomato plants and human insulin produced by bacteria for diabetics. Golden rice is also an example. This is the golden rice. It's colored yellow. It contains something we call B carotene, which is a precursor to vitamin A. And then milk can also be an example. It can also be considered as an example since it contains spider silk protein, which is produced by goats, and spider silk is extremely strong. So these are examples of genetic engineering. We'll be talking about the benefits of genetic engineering right now. Mahdi?
So basically, vaccination generally involves injecting weak, live, killed, or inactivated forms of viruses or or their toxins into the person being immunized. Gene therapy is the genetic engineering of humans by replacing defective human genes with functional copies. Agriculture benefits of genetically modified plants. First is improved nutritional quality. Second is better nitrogen fixation. Third, disease-resistant plant, enhance efficiency of mineral reuse by plants to prevent early exhaustion of fertility of soil and reduce post-harvest losses. Basically, those are the benefits of genetic engineering.
Thank you guys so much. If you have any questions, you guys can contact us. Our numbers
And
and emails
the
will be
video is
below.
left in the description, so make sure you check that video that's also related to genetic engineering.
Thank you.